Welcome to WEP. Once upon a time, a program called Winter Enrichment Program started in January 2010 at KAOST. I'm Marie-Laure Boulot, responsible for WEP since the inception, and we are glad today to welcome you. Please join me in calling on stage Dr. Tony Chan, our president.
Thank you. Thank you. I think I can walk around. Good afternoon, everyone. Welcome to WEP. You are in for a treat. And I look at the program. I met many of the plenary speakers, the variety of topics they will cover, and their expertise and their knowledge, I think will benefit all of us here. It's my first WEP. You know, I missed last year's. So now I know a little bit what I missed from last year. I think WEP is a unique experience, certainly for cows. Probably around the world, there are not too many universities with a WEP-like program. You know, where you have the period between our commencement and our beginning of class. There's this 2-week period where we try to pick a topic. We bring in some of the best people from around the world to speak about that. And the idea is not going to be just what you do in the regular classes. Right? There has to be something a little different, something that has an element of creativity, something that is a little bit out of the box. That is all involved in it. Something that involves science and technology, but also something that involves the arts, for example, the creative arts. So the idea is, from an educational perspective, is to reach out from your comfort zone, especially from the students. And so this hopefully this experience will stay with you. As you know, this is required, it's mandatory of all our students here. You have to attend a full WEP before you can graduate from KAOST. And there's a reason for that. We have, as you heard from Marie-Lou, that this is the beginning of the second decade of WEP. You know, we've done it for 10 years. Every year there's 1 or 2 faculty who we charge to be the chairman, chairwoman, to chair the committee. The theme is picked. This year's theme is personalized medicine. I remember last year's theme was time. In fact, it was first time. I mean, Time Magazine, time, no, time, in every aspect, okay? So personalized medicine, I think, I don't have to explain to you, is the wave of the future. I mean, the way I understand it, everybody, we care about medicine, right? We all eat ourselves, our loved ones can get sick. Personalized is sort of the new wave. Of course, all medicine in some sense is personalized. You get sick, it's personal, right? Your treatment is personal. But I think the new term is now you can personalize to a much greater degree than before, because we know a lot more about the genetic, the basic chemical, genetic, biomedical background for medicine. And this is especially reinforced by the digital revolution. So you combine this together with big data, the way you deal with medicine, the way you diagnose, the way you treat is completely a new world. Just as an example, I was reading in the newspaper yesterday about a report from, I think, the US about cancer survival rate, I remember. And it has been dropping dramatically. And that's because of medicine, personalized medicine, but also public health policies. You know, like non-smoking, you cut down on the lung cancer, for example. So I think at KAOST, this is particularly suited. We are particularly suited for a topic like this because we are science and engineering, even though we don't have a medical school. We have many people whose research, many of our faculty and research scientists, the research is tied to personalized medicine. We have people who study genetics, people who study engineering. Biomedical engineering is a new program we have. Personalized medicine requires a really multidisciplinary research approach. okay, from computer science, studying big data, artificial intelligence, to chemistry, to many aspects. And so, so this is what CAUS is about. In fact, some of you, I'm sure, your own research interest in this area. I want to, first of all, acknowledge and thank all the invited speakers who have come from afar. Many of them come from afar. Many of them the first time in the Kingdom of Saudi Arabia. So I hope they will have some time to get to know CAUS, but also beyond. For example, the snow in Saudi Arabia. As you know, many do not know this, okay? So there are many things that are interesting. Please, especially the student, make sure you don't just listen to the lecture, talk to the speakers, as they will be here for extended, not just one day. I also want to acknowledge the organizing people from CAUS. We have Mohammed and Naveen, they're somewhere here. Our two faculty, and Marie-Laure, you just met. The many will be beyond the three of them who are involved in putting this together. So without further ado, let me bring Dr. Najjar Ashri, who will introduce our first speaker, if I understand. Najjar? Yeah. Thank you.
Thank you, Tony. Thank you, everyone, for coming and for joining us today. I'm honored to be here introducing our keynote speaker as we begin CAUS' second decade in discovery and innovation. We are at an important stage in the university's development and indeed in the kingdom's transformation. We are privileged to have you with us and to share with you some insights of this interesting and exciting journey that we're going through. As no doubt will become evident throughout the day for those visitors from outside the university, CAUS places an extremely high value on lifelong learning and inspiring the next generation. Our commitment to creating positive change for the world and for the kingdom has been at the heart of our mission since the beginning. WEB is a reflection of this vision. It's an annual celebration of thought leadership in which we welcome the world to share our vision. I have often said that WEB is a microcosm of CAUS. Like the university itself, we bring together Saudi Arabia and the world's most inspirational change makers who are working towards common goals for humanity. And while science, technology, talent, innovation lie naturally at the core of this mission, our work here is supported and in fact supports rich cultural, artistic, and social endeavors that help people live fulfilled and productive lives. For Saudi Arabia particularly, WEB represents CAUS' commitment to ongoing and holistic learning and problem solving in action, starting here at home. Over the next 10 years, Saudi Arabia is making an historic investment in healthcare and medicine. As a nation, we aim to not only improve health outcomes, medical infrastructure, and general well-being domestically, we also want to be a global leader and a hub of expertise in breakthrough technologies, theories, and treatment for humanity. CAUS has embraced this mission and is working closely with collaborators in the kingdom and around the world to push the limits of what we know and we can do in medicine and healthcare. For CAUS, breakthroughs under this theme come largely at the intersection of technology and health. Our researchers work across scientific fields and work frames to create a special new brand of knowledge for the future. This year's WEB's theme embraces this knowledge in the form of personalized medicine and all that it promises for the kingdom and the world. With that, I can think of no better person to usher in this year's program than Her Royal Highness Dr. Maha bint Mishari bin Abdulaziz Al Saud. Dr. Maha has long been a champion for transforming the health and medicine landscape of the kingdom. She's a doctor of internal medicine, a senior clinical scientist at the King Faisal Specialist Hospital and Research Center in Riyadh. She serves at Al Faisal University as an associate professor of medicine and the vice president of external relations and advancement. At Al Faisal University, she spearheaded the integration enrollment of women's education back in 2011 and continues to be a voice for women's empowerment in the kingdom through education and entrepreneurship. As Saudi Arabia assumes the G20 presidency this year, Dr. Maha has been called upon to serve as the co-chair of the G20 think tank engagement group, the T20, Task Force on Migration and Young Societies. Under her leadership, this working group aims to explore youth unemployment, cross-generational engagement, policy development for the better integration of migrants, and increased access to education for refugee children. Dr. Maha is also a great supporter of culture, entrepreneurship, and the arts, as demonstrated in her personal activities, from hosting fundraisers celebrating Saudi Arabia's top poets, to holding auctions for Islamic art, to collaborating with the Women's Initiative Award, which support female entrepreneurs. Finally, Dr. Maha has always been a close partner and advocate of COWS since the beginning, offering guidance and insight at key points throughout our young university's journeys to what it is today. We thank Dr. Maha for all that she has done and continues to do for the kingdom, and are delighted to have her with us here today. Please join me in welcoming Her Royal Highness to the stage.
Thank you, Najat. Thank you. It's my pleasure to be here. بسم الله الرحمن الرحيم والصلاة والسلام على سيدنا محمد وعلى آله وصحبه أجمعين. Thank you so much for having me here. It's such a pleasure always to come and visit KAUST. It's really a beacon of knowledge. It's the jewel in the heart of Saudi Arabia. And at this stage, I always like to remember King Abdullah, because he is the one who initiated this, and he asked us always not to forget him when we pray, and pray for his mercy on his soul. I'd like to speak today about personalized medicine, and in that, I'd like to bring for you my personal feeling about it, because I am an internist, and this will take you through the background of personalized medicine, some science about it, and then I will discuss the genetics basis with which I would like to advocate today. Also, we cannot speak about personalized medicine without talking about personalized oncology and what it has done to precision oncology, what it has done to change the landscape. And this is also specifically for my country, so I'd like to give a little bit of flavor about Saudi Arabia in this. I will also mention a little bit about policymakers, what they have been doing regarding precision medicine, and then take you a little bit into the future. So the father of medicine, Sir William Osler, said, if it were not for the great variability among individuals, medicine might as well be a science and not an art. By the 20th century, clinicians had already developed an understanding of the association of genetics and disease and the concept of personalized treatment. They also looked with the 21st century, with the initiation of the human genome sequence, they looked into what other evolving applications were coming out of that. And I'll take you through that time trip, and you can see that they have the progression of knowledge that is taking us to this point of precision medicine. This could have not happened without a very important thing, which is technologies. These technologies are four, basically. I mean, if you look at them, they're actually 25, but you can group them into groups of 4. So technologies for greater molecular level characterization, which is basically genomics, pharmacogenomics, and all what it entails. Then we have technologies for personalized therapeutic interventions, like stem cell therapy, robotics, and so on. And then we have technologies for personalized disease and health monitoring, which is the wearables, the sensors, and so on. And then we have underpinning and enabling technologies, like machine learning, big data, and Internet of Things. In 2001, just to give you a little bit perspective, sequencing one genome cost $100 million. Now, less than 1,000. So the definition of precision medicine is an emerging approach for disease prevention and treatment that takes into account people's individual variations in genes, environment, and lifestyle. Let's look at this slide. This is the percentage of the patient population for which a particular drug in a class is ineffective on average. At best, 38%. At worst, 75%. Let's talk tumors. Here, the percentage of patients whose tumors are driven by certain genetic mutations that could be targets for specific drugs by types of cancer. You can see this. This takes us to pharmacogenomics. The basis of recommending a drug and its dose to an individual patient is the discovery of genetic variants and association with varying response of drug. So genetic makeup-based prescription design implementation therapy will improve the outcome of treatments and decrease side effects, and a better understanding of individual variations and effect on drug response, metabolism, excretion, replace the one-size-fits-all and trial and error. So here, what you do in precision medicine, you take a patient population, you apply pharmacogenomics tests, you get your therapeutic biomarkers, and you apply them to genetic variants of the individual, and then you can decide what you do with the drug. This is fantastic for patients because it will increase the opportunity to prevent the disease, improve quality of life, help avoid adverse reactions, and so on. Also, it will benefit the healthcare system. So you more quickly target the right treatment for a patient, you have the optimal dose, and you can focus efforts on prevention and earlier detection, and improve access to clinical trials, which is important. Biomarkers, this is important. Biomarker is a molecular, biological, or physical attribute that characterizes a specific pathophysiological state and that can be measured and evaluated objectively. This is very important. This will help you identify or define progression of a disease or predict and quantify a therapeutic response. You can attach to this biomarker. You will find out if there is a risk of developing a pathology, presence of the pathology, risk stratification, and measuring response to therapy. These biomarkers will have prognostic versus predictive distinction. The prognostic is when you provide information and outcome independent of therapy that is used. Predictive provides information on outcome with regard to specific therapy. This is important, and we have seen biomarkers like HER2 in breast cancer and Wrasse and KRAS in colorectal cancer. So, can we identify and clinically validate biomarkers which will improve the precision of the 4 R's of cancer medicine? Yes. It will help us in the prognosis, which is selecting the right patients who will benefit most from the treatment. In prediction, we select the right drug or the combination of drugs. Safety, we'll select the right drug dose. Risk, susceptibility, we can select the individuals most at risk of developing a specific disease or for intensive screening. So here, what we can do, it will change the risk-benefit ratio of chemotherapy. With using these biomarkers, what you can do is you can identify patients post-surgery. If there is no burden of a disease, and you know that they are low risk for recurrence, you will not give them chemotherapy. Otherwise, if they are at high risk after the surgery, you can give them chemotherapy. So this is a new paradigm. This is when you don't have personalized medicine, you try and error. If you do have personalized medicine, each patient will receive the right medicine for them. You can apply the biomarker diagnostics, and you can stratify your patients to some who will have therapy, some who won't, and you can decide who benefits from what. This is an example, which is the Wrasse testing for colorectal cancer. If you have that gene, it means a certain drug that is very important in colorectal cancer is resistant to it. So it's very important to determine at diagnosis of that cancer whether you can use that drug or not. So if you have the biomarker saying you have Wrasse, you cannot use it, you have to employ other interventions. Colotox works in a different way. It's a biomarker which selects the right drug dose. So it's applied where you can find whether this drug is going to be toxic for the patient, and maybe you can decide on an alternate treatment. What's new? Liquid biopsy. This is a very exciting field, and this is where you have the tumors having secreting tumor cells, releasing DNA, and circulating tumor cells. What you do is you extract them from the blood. And this is fast and less invasive way. So this is one of the innovations. The second thing is pioneering genotyping technologies will enable more personalized medicine. So you move from the current, which is single gene-based targeted therapy, to multiplexed molecular tests. Let's see if I can take you through genetics and you liked it. I'm trying here to advocate for more towards focusing and resource allocation. And before I say that, I want to show you what medical errors do. What do 1,100 jumbo jets and preventable medical errors have in common? Any idea? If you crash 1,100 jumbo jets, the amount of lives lost are exactly the same amount that we are losing for preventable medical errors. We don't accept that, right? We don't accept one airliner to go down, right? And it doesn't happen because we do know how to build planes, we do know the technology, we know what to do. This is what we are aspiring to do with cells, know exactly what's happening with cells. And we are on the way. Here in a lab, they created a bacterial cell controlled by a chemically synthesized genome. 1 second, please. Coming from a country where consanguineous rate is 58%, and mostly 60% is to first-degree relatives, we have an awful burden of genetic disease. So when we speak about resources and we speak about allocating research, so what we're seeing now is Mendelian diseases, they are not being targeted for research as much as diabetes, coronary artery disease, hypertension, and so on, what we call complex diseases. Well, let's go to our Middle Eastern wisdom. Ahikar told his son, a sparrow in your hand is better than 1,000 sparrows flying. Let's see if we can apply precision medicine to Mendelian diseases. These are the 4 Ps of precision medicine: predictive, preventive, personalized, and participatory. What's common in this slide is each picture here depicts one genetic mutation causing this syndrome. So each one of these patients has one genetic mutation. So it is precise. So if we know the genetics, we know exactly what to expect. What we do right now is we see the disorder. This is the approach. Then we suspect a diagnosis. We sequence one gene. If it is negative, we reassess again. If it is positive, then diagnosis is confirmed. This is lengthy, costly, and inefficient. If you apply clinical genomics, diagnosis is confirmed right away. Let's see what they've done at King Faisal Specialist Hospital. This is from a study they just published. Actually, what they've done is they've asked all the clinicians in the hospital, any patient you have, you can't understand what's their diagnosis, you have not reached the diagnosis, you're baffled, please refer them to us, and we will do whole exome sequencing for them. So 2,219 were involved, and the age was from fetus up to adult. We had 322 adults. 274 clinicians actually contributed from different subspecialties with a very broad range of primary indications. So some of their patients had deafness, some had developmental delay, or liver disease, immunodeficiency. What was the yield? It was interesting. 56.9% were solved. They had a diagnosis. And the correct diagnosis was not suspected in the majority of the solved cases. We also were able to identify in that study all these genes that were associated with these symptoms. I'm not going to tell you about these, but it shows you that you can have both two diseases in one patient. So the next thing is predictive. So if you look here to the left and you apply perinatal or pre-implantation testing, what you're going to have is, let's say you stratify them, and you're going to see that they have high genetic risk of coronary artery disease, as in this study. And you go on and you know that this is what they have, and you apply a favorable lifestyle. So they cannot smoke, they have to live a healthy lifestyle, eat good, and you have actually decreased their genetic risk. Their probability has gone down. Even the genetic risk is the same, but it will go down as if you are living an unfavorable lifestyle. So it is predictive. And it is preventable, because, you know, if you look at the genetics and you know the mode of transmission, and what you can do is you can offer pre-marriage counseling, and what you can do is you can prevent this. And we think they're common because there's plenty of them. If you add them all together, there's plenty of genetic diseases. This is the burden of autosomal recessive diseases in the community. Please don't forget the landscape of consanguinity that we have. One more thing that is important that we want to stress, that it is very helpful with studying Mendelian diseases, is because they are functional annotators, the functional annotators. Let's look at this study. This is a study which was done on mice a few years ago and published. In it, they postulated that there is a mutation in this specific gene, MICU2, that will cause a certain issue with the cardiovascular system. Later on, this study, when the same mutation was studied on a human being, on a person, they found out that the deduction that they had from mice was not true, was not the real thing. And it was published later on that that gene causes something else totally when you are studying in a human being. And it's because they missed the signaling. So, are there real divides here? Is there Mendelian and there's complex disease? Is this a real thing? Are they two are very separate? We don't think so. We think they are blended. And what tells you this is skin tone, right? So skin tone varies all over the world, and we know that pigmentation is highly heritable and polygenic. So if you lose one of those genes, that TYRP1, you end up with albinism. What we have done also is established that there is an association between complex diseases and certain genes. So here we have established that with ADHD, myopia, neural tube defects. And if you look at Greenland, they have one specific variant that causes most of their type 2 diabetes. Also, there is a specific variant of genetic mutation that will cause familial form of systemic lupus erythematosus. We've also found that out in obesity. Obesity, which is a huge problem in Saudi Arabia. 34% of our population are obese. This will take me to a very important thing that I want to mention, which is druggable genome. The druggable genome is 130 families, 3,000 genes that are that express proteins that can be altered by drugs. So this is a frontier for treatments, for devising drugs, creating drugs. Why? Because now to do any trial, so you can manufacture a drug or discover a drug, only 4% will go on to become, you can sell them. What we have now are different gene therapies that are being used. This one, Luxturna, was just used a few weeks ago in Saudi Arabia for a patient. This is $425,000 injection per eye. This is more genetic therapy that's been used. And I'd like to mention cystic fibrosis. They've done a good breakthrough therapy for them with the genetic diseases. This is Dr. Fozan Lakraya, whose work on genetics at King Faisal Specialist Hospital. He had discovered more than 400 novel genes, and he's still going. Again, I can't speak about precision medicine without speaking about oncology and how it changed the landscape. This is Dr. Jazia, who was great. I'm grateful for him because he shared this slide with me. This is his forecasting for cancer progression in Saudi Arabia. This is what we think is going to happen. Precision medicine has benefited patients all across, but mostly, as you can see here, hematology and oncology. What we usually do as physicians at this point in cancer is we rely on histology, location, size, and limited by only 3 modalities of treatment and limited supportive care. But if you look at what's going on, since the 90s, you'll see that survivorship has been going up and mortality has been going down in cancer. And why is that? Because all of precision medicine and targeted. So if you look at these two charts, between 2003 and 2013 here, you will see that we have changed, decreased a little bit of the cytotoxic treatment, and we have increased in the targeted medicine. So we identify and target genomic alterations, and that changes the narrative. Is that simple? No, because there's plenty of mutations in human cancers. So far, there is a big change. So if you look at chronic myeloid leukemia, introduction of this drug has tripled the 5-year survival rate. And if you look at metastatic melanomas, which is a very aggressive disease, 5-year survival is 15 to 20% only. Identification and mutation in the BRAF gene has guided us into specific therapies. Now there are 3 new therapies that are improving lifespan. In metastatic colorectal cancer, also the Wrasse and KRAS mutations, if you know you have to adjust your treatment, but it has to be at diagnosis. But there is an improved survivorship. Herceptin in breast cancer is also another focus for treatment. What about policymakers? I'm sorry, this is more cancer stuff. Well, personalized medicine has been on the horizon for everybody, for many people. They are looking at it and they are implementing it. If you look at the United Kingdom, they have just finished the 100K, which is they have taken 100,000 people with rare diseases and common cancers, and they looked at them and their families, and they sequenced them. They've done that also in the United States. At the VA hospital, 800,000. There is also more work with the European countries. There is the International Consortium, which also has leading a great effort into precision medicine. In Saudi Arabia, we have the National Program for Gene Sequencing, and we have also other efforts. So what's next? There is customized vaccines, and there is the CAR T-cell therapy. CAR T-cell therapy is interesting because what you do, you collect these cells, and you genetically reprogram them, and then you grow a lot of them, and then you reinfuse them to the patient. This is a favorite of mine. It's called the Amazon Bank of Codes. This is an open digital platform that maps biological assets and codifies rights and obligations related to their use. And it's called the ABC, so it looks to enforce the Nagoya Protocol, which is an international convention to promote fair access to genetic data. There's also nanomedicine, which targets genomic changes that cause the cancer, and you engineer nanoparticles loaded with a chemotherapeutic agent targeting the mutations. And these are drugs that are going to be, FDA approved and available. So we have diagnostic and prognostic. You have a third one? Yes, we do. It's precision prevention. This is a new concept evolving right now. And it's actually, the goal of it is to target right intervention to right population at the right time. Here we go. So I think there's a lot of collaboration that we can have between KAUST and King Faisal Specialist Hospital and Al Faisal University, where I come from. So Sir William Osler said, don't tell me what type of disease the patient has. Tell me what type of patient has the disease. And I'd like to finish by thanking all the people who helped me create this talk. And I'd like to thank you for your listening to me. And thank you for having me here.
Thank you very much. It's really a great pleasure for me to be here today. I cannot imagine any speaker being happier than following Her Royal Highness Maha bin Mashrahi Al Saud. This was an incredible talk that really introduced precision medicine and personalized medicine the best possible way. And it makes it so much easier for a scientist to continue, having heard such a great medical scientist talking. First, I would like to thank the organizers of this particular conference, the leadership of KAUST, President Tony Chan, my dear friend, Vice President for Research, Yves Nanou, about whom I will speak in a little while, all the other colleagues, some of whom I know already, Naveen, for this magnificent introduction. Ladies and gentlemen, we live in a world that is changing continuously, and we owe it to our people to help them. They look at us for solutions, and the solutions have to come from us. We don't work for more publications, for more patents, for more companies, for more academies, and for higher impact factors and citations. We work because there are people dying out there, people dying from 2 or 3 diseases together, and we really need to provide a better life for them. So precision medicine and personalized medicine are pretty much areas that have become the standard in drug delivery these days. This is a slide that I have borrowed from my good friend Rashid Bashir, with whom I have worked together. He is now the Dean of Engineering at the University of Illinois. And it shows really how much we've changed. I became a biomedical engineer, I don't mind saying that, Naveen, In 1967, December, when I saw how Christian Barnard at the Hospital Gros de Sur in Cape Town did the first heart transplant. And I said, that's what I want to do. But I knew that as an engineer, I could listen to the medical doctors, listen to their problems, and try to provide a small little stone to solve those problems. The global population has changed enough in the last 50 years. We are expecting by the 2050, we are going to have 9 billion people in this world. How are we going to feed them? How are we going to treat them? People are living longer, and they expect healthier lives. At the same time, we realize that all of us work for the common good. The solution of cancer is not simply having some medical doctors working by themselves in a corner, or a molecular biologist. All of us have to come together, and that in English is called convergence. And we really need to break the barriers. And I love Coast, because barriers have been broken here. Because everything has been open and people across areas work with each other. And that's happening more and more throughout the world. It's happening in the United States. It happens in my university, which is one of the places in the forefront of this convergence technology. And when I look really at the future, I remember very distinctly 30, 40, 50 years ago, what electronics and computers did for society. And I believe it's time now for the same thing to happen with medicine. But the costs in healthcare are unimaginable. They are unsustainable. I happen to know the market in the United States better. Look at this particular curve. Every 10 years, we double the price. But Europe is not very much behind. Japan is not very much behind. I have no data for Saudi Arabia, but I believe that there is a continuous increase. So we have to address this problem. At the same time, we have to provide a higher quality of life. Why? Because we cannot have the patients forever be treated the way they were being treated 50 years ago. We cannot have patients take tablets and capsules and expect that that will be a relief for the tremendous pains that they have. This is a slide again from Rashid Bashir, but at the same time, it addresses something that I have in the bottom. I highly recommend you find this paper in Science Advances, a journal that I happen to be one of the people editing it, although this, I did not run it myself, somebody else did. And it talks about system network theories and tunable responsiveness of precision medicine. How you can really modify the properties of a particular system in order to adjust it for a particular patient or for a group of patients. Really a tremendous problem. It has received really accolades and many comments and so on. The idea of convergence is not new. In 2014, at the National Research Council in the United States, several of us got together, we sat down, and you can see Joe DeSimone and Cato Lorenzin, major figures, 3 academies of the United States, all of them, and we came up with this book. Highly recommend you find it. It's free, actually, I believe. You can get a copy and read it, what Convergence has done for our field. And Shu Chen, Rashid Bashir, Bob Nirem, and Rod Pettigrew got together and they said how this translates into what happens in the biomedical field. Translational medicine. This is another word that has become very important for me. You see, my generation, I don't mind telling you, I'm 71 years old, going to 72, but hopefully I still have a heart of a 40-year-old. And I can tell you that really what we were doing 50 years ago is science because we loved to do science. There's nothing wrong about it. But who is going to pay for it? Do you really expect that the taxpayers somewhere in a poor village who they don't have anything to eat will be paying taxes so that you and I can write our publications? No, that's not the future. The future is coming up with solutions that are translational. That is, they pass to the patient relatively fast. Please, especially to the young generation who are probably up there, make it a point in your life to help your country, to help your continent, to help the world, to help the poorer that don't have anything else except you to look at. And I believe, and Rashid believes, that there's going to be a significant change also in education. The days where, as engineers, we were always looking at medical devices and so on are gone. There has to be a strong incorporation of biology in it. And I apologize to my medical doctors. I expect that you need to spend some time taking a serious course in physics or a serious course in chemistry to really better appreciate what this nanotechnology is all about and so on. Now, as I continue, there is something I want to say specifically about specific examples that I want to give you today. When I arrived in the United States in July of 1971, on a very hot and humid day, I was coming to study biomedical engineering and chemical engineering without knowing what that was. And when I started working in the field, I was working with materials and systems and biomaterials and so on. There was no drug delivery yet, that existed already on the shelf. They had been tested a little bit for toxicity, but not very much. The world has changed. That's not how we create new drug delivery systems, new biomedical systems, new biodevices. We have advanced biomaterials. We know very well how specific components of a biomaterial will affect the properties and the interaction with the body. At the same time, we have biological materials that are modified or hybrids. They contain a proteinic section and a polymeric or a synthetic section. We have systems that give intracellular delivery, because as I will show you in a few minutes, there are diseases where we would like to deliver the therapeutic agent inside the cells without affecting the cells otherwise. We have new nanotechnology and bio-nanotechnology techniques. And in the long run, I tell my students, I tell my own biological children who are relatively young, 19 and 16, I want, by the time I go to another world and you take over, I want to be able to see that we have a system, a device, a product that can be embedded inside my body and can recognize the beginning of a particular disease and can send the information to a patient and at the same time can treat the disease. Is that difficult? Well, we'll see in a few minutes, but here at KAUST, I'm talking to people who have this background. You have exceptional computer science programs. Your own president is a leader in the field, and he's a member of the National Academy of Engineering. This is really the type of empowerment that you can have in the field to say, I will do something different for my people. In the last 15 years especially, intelligent biomaterials and nanoscale biomaterials have become very, very important, intelligent. What is unusual about them? Are they smart? I'm about to show you that they are very smart. In my laboratory, we work with all types of materials, especially those that are called gels, hydrogels, that we have the ability to modify by changing the pH, by changing temperature, by changing electrical properties, by applying ultrasound, and so on. So basically, the benefits will be that we will have systems that can be triggered by the surrounding environment. Think about it. When I talk about pH, that means I could take something orally, let's say, that would be at a relatively low pH, and it will not interact. And then when it gets in my stomach and be at a higher pH, it will expand, and it will start releasing. This is a very simple principle of the idea of drug delivery in a smart system. So smart means thermodynamically interactive. It means that there is thermodynamic with the system. These gels, these hydrogels, are not something new. If you really know history of chemistry, you go back 100 years ago, you're going to find them. This year we are celebrating the 100 years from the day Hermann Staudinger in Germany used the term macromolecules. And probably you know the story that in his first papers, he was rejected immediately by people saying, well, dear colleague, it is well known there is no such a thing as a macromolecule. These are colloidal structures. That's all they are. He was not the only one. Paul Flory in the 40s was the one who really taught us how to put together biomacromolecules and regular macromolecules. And it was in the 50s the Soviet Union, especially Nikolai Plate, and in Japan, Ishiro Sakurada, who is on the very left side. And then it was in 1959, somewhere in Europe, in Czechoslovakia, in Prague, that Otto Wichterle presented the first material that was non-toxic, non-carcinogenic, and could be used for contact lenses, soft contact lenses. I was lucky. I went to MIT at a time when all these things were happening, and I met this man who happens to be a Philolen. The first time he talked to me, he started speaking in ancient Greek, reading the Iliad of Homer. This is another story that I will not talk about. The reason I'm mentioning that name is because there is some connection with this world. I did my PhD with him. He came very often to France. He met Paul Remp. This is a book they wrote together. And of course, Yves Nanou, the vice president of research here, is Paul Remp's former student, came to MIT, worked with Merrill in the 80s. And on the very right side, you see Professor Merrill at the age of 97. I have a wonderful video that I showed yesterday greeting Professor Nanou and me on this occasion. So really the world is still there. Why is he important? He was the first one that in 1979 said, you know, polyethylene oxide is unusual. Polyethylene glycol is unusual. Under certain conditions, it can be used as a stealth material. So I can create nanoparticles that can go in the body. They will be circulating, and they will not be recognized by the reticuloendothelial system. And that led to this explosion of pegylated systems. Yves Nanou and I and many others have worked with these systems for a very long period of time. In the remaining time today, I want to share with you a few ideas about certain advanced systems that are intelligent, as I like to call them, oral siRNA delivery, oral delivery for interferon beta, and I want to talk also a bit about detection. In fact, why don't I start with the detection? I want to see someday nanoparticles that I can take by some method, orally or by injection and so on, and they will be able to recognize that something has happened in the biological system that will be a true personalized medicine and respond to that system. And look how I prepare them by creating these very nice nanoparticles on top of which you have these tethers. And the tethers have been taught, intelligence, they have been taught to recognize one particular compound, green. For example, if I want to treat insulin diabetes, maybe they can recognize glucose. They capture the glucose, they start releasing insulin by a process that eventually will lead to an in situ delivery of the insulin. And with several of my students, these are two of the most capable. They are both postdocs now, Heidi Calver on the left and John Clegg on the right. We have come up with new drug delivery systems with rather complex structures, if you can see. We can really put all kinds of targets in them. You can put all molecular structures. Or on the left side, we can have sensors, systems that can sense that a particular disease is starting. And this has led to the idea of molecular imprinting and molecular recognition. I see some of you leaving. I hope something terrible happened in your family, because we are getting in the best part. On December 25th, 1992, I started not seeing well. Double vision, the doctor said. I had no idea what it was. A few weeks later, the diagnosis came. I had a serious autoimmune disease called multiple sclerosis. This particular disease affects the myelin on the nerves. The result is inflexibility of the nerves. The result is I cannot see. I cannot walk. I feel like I have ants on my legs. There are huge problems. There is incontinence, and there is impotence. I was newly married. We didn't have children. As you can imagine, it was a major problem at home. Now, all of us, whether we are medical scientists or engineers or scientists, we always look at the problem. We never look at the effects. And we are here to address that all of us have to look at the effects. What do we do with patients like this? I started working on the development of certain systems for delivery of interferon beta, which is one of the main components for treatment of multiple sclerosis. I will tell you about it in a little while, but in the process I learned that in multiple sclerosis, in the very, very early stages, there is osteopontin produced in the CNS system. If there was a way for me to measure the osteopontin continuously, I could perhaps send a signal to the doctor's office. The concentration of osteopontin is elevated, therefore bring the patient for additional studies. Therefore, the MRI, the magnetic resonance imaging, would have to be done much faster than in my case, which was done 6 months to a year later. I don't want to keep you suspended, although I will come to this situation. 15 years later, it was discovered that I had been misdiagnosed with multiple sclerosis. I don't have it. You can see me walking still. I have another autoimmune disease that is easier to control, but I present this as an example of really what you can do, what a scientist can do when bad things happen to him. With these wonderful students, both of them becoming faculty members now, John Clegg and Marisa Wexler, we were able to create structures that have these nanovacuoles, and the nanovacuole can capture just a particular compound, the osteopontin, glucose in the case of insulin, and trigger a release and start sending some small amount into the blood, and at the same time doing, of course, informing the doctor that something has happened. Look at this nice system. On the left, I have a fluorescent compound containing glucose, which can be seen because it is recognized by the particles. On the right side, I don't have any recognition. This is the type of process we can come up, and now we can recognize even large molecules. Large molecular weight molecules, proteins and other compounds like this. Now, you're going to say, where are the possible applications? Again, Heidi Calver and another one, Stephanie Steichen, who is working now with Dow, DuPont, that is, were the ones that started looking at the possibility of recognizing several proteins at the same time. And this process led to creating new type of gels where in the pores we had specific systems that could recognize just these particular compounds. What are the applications? Frankly, there is a disease that 6 years ago I had no idea what it was. The disease is called Sjögren's syndrome. It's a disease that appears usually in young girls at the age of 6, 10, 12. They start having dry mouth, dry eyes. You have a dry mouth, drink some water. You have dry eyes, put some, I don't know if you have Visine in Saudi Arabia, something similar. No, the disease is much more serious than that. And it is debilitating, and people die after 10, 15, 20 years. So we started working and saying, can we detect this disease? We identified 3 components that were very specific in that particular disease. And we used these 3 components to create these sensors with the little nanocavities in them that could capture these 3 components. And we have enough studies now to show that if you capture the 3 components, you have a very high probability to identify Sjögren's syndrome. Not with 2, not with 1. If there is a 4th or a 5th one, perhaps. We are working on a variety of diseases right now. This work has been published by Heidi Calver, and there are patents. And it shows you really how you can use a biopolymer and the drug delivery ideas to recognize the codes for the particular process. Here are the 3 compounds on the orange, pink side. You can see how we can really recognize these systems. Now, I come to something that is very important. Please look at the paper in the very bottom. This is a paper in Progress in Material Sciences. It's called Modular Fabrication of Intelligent Material Tissue Interfaces for Bio-Inspired Biomimetic Devices. This is a very important field for us. This paper is 50, 60 pages, has 600 citations in it, references in it. And it is a paper in which we expose to the world everything we think about this field. And one thing that I think about this field is artificial intelligence. I gave you already a simple idea of how you can have a nanoparticle or a nanosystem that has the ability to recognize a surrounding fluid or a surrounding compound. The question is, it can recognize it. How can it treat it? Typically, you will say the treatment will be some drug delivery. I'd like to be able to control it. Can I have some small nanorobots incorporated into that system that have the characteristics of artificial intelligence so that I recognize that there is osteopontin here, and now the nanorobot takes over and says, under the conditions of recognition, this is probably the disease. And it starts immediately. This is the black part that you see here, how you can incorporate artificial intelligence in those systems. All of you have heard of AI. And you know that it is a magic word. Doctors like to use it, scientists like to use it, frankly, sometimes even ministers of education and health like to use it. Artificial intelligence means that if I give a system certain characteristics, the system has the ability to respond and treat a patient in a particular way. But this, you have to have hundreds of thousands of data incorporated into this nanorobot in order to do this analysis. This is where our field is going. Will it be a product for tomorrow? Absolutely not. And I can tell you, in a litigious world, like some of the Western world is, there will be, I'm pretty sure, situations where people will try to say that a particular device like this is responsible for something that happens to the patient. We are always very careful, all of us, but this is one of the futures of the system. The other one is the idea of coming up with systems that are stimuli responsive. pH affects them, temperature affects them, and many other characteristics. There are several papers. I don't mind leaving the slides here for anybody who would like. Naveen, you can take over and give the students the slides so they can go to some of these references. One area that is particularly important to me is the solution of the problem of multiple sclerosis. And not only multiple sclerosis, other autoimmune diseases and diabetes. What is my problem? I believe that patients do not like injections. They don't like shots. They prefer something that they can take orally or something they can take transdermally. Highly recommend to you, listen tomorrow to the talk by Professor Zhen Gu from the University of California at Los Angeles, who will tell you about an incredible new method of delivering insulin through the skin, something that 30 years ago we couldn't do it. Anyway, coming back here, in my case, I'm interested of how to deliver these proteins through the intestinal wall. Rupture the intestinal wall, find paths in between, or something like that. And I do that with several techniques, one of which I will discuss with you, an externally responsive nanoparticle later, and the biologically triggered particles, but on the very left, and I will conclude with that, also with theranostic systems. Theranostic systems are really another form of personalized medicine. So let me tell you very, very fast, because we don't have much time, what I work with. I want to treat different diseases with oral protein delivery, because I am convinced that we need to have systems where the patient would be willing to take the therapeutic agent. If you tell me that the patient is going to have 4 injections a day, that is not a solution. The same thing with calcitonin delivery for postmenopausal women, growth hormone delivery for dwarfism, interferon beta delivery for multiple sclerosis, and delivery of factors 8 and 9 for, what do you think? hemophiliac disease. All these poor little boys that every day or every second day they have to take the injections in order to put back the factors that they are missing. Otherwise, they will die from bleeding. And all the mothers that have this stigma that they are responsible, that the gene transferred to the little boys. I happen to be the luckiest man in the world that I'm married to somebody who is also a biomedical engineer, internationally known, Dr. Lisa Brannon-Pepas, who was the first one who came to me about 10 years ago and said, Nico, do you know what's happening with hemophilia? Of course, my PhD was on this subject. No. What happens to the parents? And that was the first time I realized what was happening. The first time we came up with a hemophilia factor oral delivery product for factor 9, you cannot imagine how many mothers called me at the University of Texas to ask when it would be available for their boys. And I realized what a major problem we have. Very fast. The problem is serious, ladies and gentlemen. I pulled out this map from a previous talk. This was a prediction of the World Health Organization in 2006 about what was going to happen 2025, that is 5 years from now, in the world with diabetes mellitus, where you see the darkest color, that's the worst case. Are you careful? Did you see which country appears there? The first time I saw it, I did not know. I said, no, this is a mistake. It cannot be Saudi Arabia. And yet it is Saudi Arabia. And in my interactions the last 2 days with several colleagues, I have gotten to realize that there is a problem with obesity and with diabetes mellitus. So let's be serious. We have serious problems throughout the world. Not that the Americans are better, okay? We eat a lot and we kill ourselves. But it is characteristic of what we as engineers, as scientists, have to do. Sit down with your medical doctors, and I always show Princess Maha on the right. Sit down with the medical doctors and come up with solutions to the very important problems they have. In my case, we were able to come up with a solution for oral delivery of insulin. We went and we studied very carefully the microvilli, the tissue. We realized we had to go in between the microvilli. We had to go inside the cells and get on the other side of the blood. We do that with a system, for those of you who are polymer scientists, that is a polymethacrylic acid with polyethylene glycol. It hydrogen bonds. It expands and contracts. Guess how? In the esophagus and the upper small intestine, it's collapsed, and it protects the protein. In the lower small intestine, and especially in the stomach, or in the lower small intestine, it expands. So as soon as you pass the pylorus, it expands. And you can start having the release. On this platform, we have modified it now. We use it for a number of different compounds. On the lower right side, you're going to see insulin. You're going to see Humira, which is the industrial name of a very important antibody that is used for treatment of Crohn's disease, another very important autoimmune disease. And of course, calcitonin. Above it, you're going to see insulin, you're going to see a growth factor, and factor 9 for the treatment of hemophilia. And this is how the transport happens. Those of you who are engineers in the audience or physicists, this is your subject. How do you transport this protein intact through the intestinal wall, in between the cells? I know you're going to say you find a way to open the cells. That's called paracellular transport. And the first time I presented it in a meeting, I don't know if Professor Li Huang was in the audience or not, but the pharmaceutical scientist said, Professor Pepas, and what about other compounds? What other compounds? You're going to be treating diabetes, and you're going to be having at the same time cholera, because you're going to be passing viruses through the openings. So paracellular mechanism is not the best. The best mechanism is, of course, a transcellular mechanism on the very left, or transcytosis or endocytosis. To make the long story short, these systems that I told you have worked very well for us. They collapse, they control the protein, and then at certain levels, above the pylorus, under the pylorus, they start expanding and they release their contents. I want you to see how when they are ready to release their contents, they extend their tethers, so they become like anchors, like a ship, and they stay on that area. And for those students who work for Professor Nanou or others, do you remember what he told you about Pierre-Gilles Dejean's theories? Do you remember what he told you that if you bring two gels together under certain conditions, they will become one gel? What paper am I talking about? Prager and Terrell, 1978, Journal of Chemical Physics. What other papers? 1990, Brochard and Dejean. That's what that is. It's the application of the polymer physics principles in a biological system. We are bringing now the tethers to really stay there for a period of time and allow the delivery of the protein. Look at the results. The protein does come out. Look at the right side. The glucose is down. This is in Wistar rats that have been pancreectomized. So you have a simulation of the real case in the body. And of course, again, another pharmaceutical scientist told me, yeah, fine. I mean, I tickle a rabbit or a rat, and the glucose concentration goes down. What is the etiology? The etiology is the protein went high. And you can use this area under the peak to calculate the pharmacological bioavailability, and you have a reasonable 20–30% biological bioavailability in these systems. And of course, you have to do that for all types of animals and effectively for humans before you get into the body. But look at the biodistribution. You can see particles now, upper small intestine, middle small intestine, lower small intestine, 1 to 4 hours. You can see clearly that you can deliver the system in the body. And I believe that nature always gives us a present, because at the same time, look what's happening here. We have an inhibitory effect on the proteolytic activity of the enzymes. Why? I think it's the first time I give the talk, and I happen to have the originator of the idea in the audience. Klaus Michael Lehr from the University of Saarbrücken is here. Klaus Michael, I always mention your name, but this time I'm particularly delighted to mention it. He was the first one. I think it was in his postdoc where he showed that the carboxyl group of these materials was responsible for this proteolytic activity, cutting down the proteolytic activity. Really magnificent work going back 25 years ago, but really it worked very well for us. One last thing I want to show you here is that very often you can bind the protein with transferrin, and this was an idea that I think Li Huang had first, and you can have it passing through the stomach. It's not released. It arrives in the upper small intestine, and look what happens. It passes now by endocytosis, and it gets into the other, or facilitated transport, it gets into the other side. This is really the future of these systems. If you look at the slope, you will see it's much higher in the presence of these systems. I'm going to go a little bit faster because I am a little bit behind schedule and there is something I want to say, and that's the application in autoimmune diseases. These diseases that I show you here have one common characteristic. This is rheumatoid arthritis, osteoporosis, macular degeneration, and multiple sclerosis. The first one is treated by Humira, high isoelectric point. The second one by salmon calcitonin, high isoelectric point. The third one, Lucentis. The fourth one, interferon beta, high isoelectric point. Much more difficult to deliver the therapeutic agent. And you know something else? You have to deliver it at a high concentration. The chief technical officer of a company, and especially the chief financial officer, is not going to accept putting in the market a product that has only 20% delivery, because these are expensive therapeutic agents. These are the characteristics of those therapeutic systems. And look how many different proteins there are. And all of them have very high annual sales. And the companies are companies like AbbVie and Amgen and Takeda and Pfizer. And these are some of the important compounds. I always show this study. This is the first study that showed this effect. Look at the very right panel. Oral delivery of interferon beta in animals, And indeed, the interferon beta remains constant. Ladies and gentlemen, you cannot imagine how important that was for me and for many others who suffer from multiple sclerosis. Once it was discovered that I had multiple sclerosis, I had to take injections intramuscularly in 6 sites, 1, 2, 3, 4, 5, 6. I would never take it up here for reasons that I was afraid the needle will break and will stay permanently in my hands. Because suddenly you're not a scientist anymore. You are a patient. I would take it down there. I can assure you, I know how to do injections intramuscularly in apples and oranges and pears, but not in myself. I would always stop and not do it. And my wife had to do it. And the day after that, the concentration of interferon beta would be very high. We are talking about very serious problems. I want you to find new ways to alleviate the problems of the patients, to make them have a life again. It's not a solution that after I take this drug for the next day or 2 days, I will be in bed suffering. That's not a solution. This is an oral delivery system. It was developed with help from the company Toray Industries in Japan, and it shows you really how, with some support, you can continue doing great work. There have been more studies on that. You can see it very nicely here with these studies, 2008, especially the right panel, and you calculate the area under the peak, and you can see you have biological activity. There is one application, which is the Crohn's syndrome. Crohn's syndrome is a debilitating disease that affects our intestinal wall and so on. There are not really many oral delivery systems. Crohn's syndrome is so difficult that sometimes the doctors decide to have an excision of a part of the intestinal wall. Okay, so a part of the gut, they would take it off. In the very early stages, you can go and treat it with siRNA. How did this start? In 2006 with Fire and Mello, who got the Nobel Prize. Since then, of course, everybody has been working on those systems. Look what we did. We realized that here it's a stomach. It's not only oral delivery. It has to come to the cells and penetrate inside the cells. And I'll tell you, I have some great scientists. One of them, Jenny Naib, she works now from the Laurel Livermore Laboratories. She had this ingenious idea to create a microsystem that I can take orally. It's ionically bound. It expands in the stomach, and it delivers not siRNA, but particles that have siRNA in them. So these nanoparticles can be delivered. They go into the cells. They open up the cells. They deliver their compound. 2 more minutes, and we finished. And they treat the patient. And these particles are made, for those of you who are polymer scientists, by a very careful technique. It's an Arget polymerization, like the ones that Chris Matriaszewski has done, and you're all familiar with. We can control, with a number of generations, what we are delivering. And these are 3 of the scientists that worked on this. From the left, Diane Forbes, and Hannah Frizzell, and Mark Reichel. And they were able to come up with the systems that they can actually penetrate. This is my last academic slide. You can see here with color confocal microscopy, where you can see with the blue, with the green, you can see the nanoparticle. With the blue, you can see the cell, with the blue, you can see the cell nucleus, and of course, with the red, you can see the cell membrane. We have penetration in the cell, we have action, and we have done knockdown studies, and we know that these systems work. As you can imagine, first I want to thank you for your attention, and I spent an extra 2 minutes, but I want to tell you the future in drug delivery and personalized medicine is really tremendous. We have to work all together. We have to come up with new systems. This is a system that I imagine, a system that can have a nanoparticle that has the ability to recognize with a biomarker that something is happening somewhere in the body, maybe in my brain. Maybe it's a system that can go through the blood-brain barrier and send that information that something is happening because it was able to, through the biomarker, to recognize the process. and start the process of delivery or of early treatment of the patient. That's where we are. I'm going to be working a few more years. I'm excited about the field. The group is large, and I want to thank all of them. It's a multinational group, all races, all ages, young age mostly. They get younger every year. I don't know how they do that. But, and of course, you see many women. I only take students who are really concerned about patients these days, and they really want to work on these problems. I believe someday we will have advanced intelligence systems that will be able to improve the health, the treatment of patients, and who knows, will give us a better quality of life. NIH, NSF, the Gates Foundation, others have supported the work. The facilities, as you can see, are like any major university's facilities, very well developed and so on. There is an institute. As I close, I want to leave you with one mark. Be excited about what you're doing. Your governments do not owe anything to you. Your taxpayers do not owe anything to you. You owe everything to the world. You became good scientists because somebody provided you the education. Give back. Thank you very much.
Good afternoon, ladies and gentlemen. My name is Donald Bradley. I'm the Vice President for Research here at KAUST. It's my great honour to introduce our next speaker, Dr. Ian Campbell. So Ian is the Executive Chair of Innovate UK, a role that he's held since May 2018. So you might ask the question, what's Innovate UK? Well, UK is the United Kingdom. Innovate UK is the governmental organisation Ian had an extensive entrepreneurial and business experience in the healthcare sector, especially in medical devices. He studied biochemistry as an undergraduate and a PhD student in the University of Glasgow in the late 1980s through to the mid-1990s. He then spent a decade working as a technical lead, then a business development lead in medical diagnostic companies, including Biocore, Corning Microarray Technology, Automation Partnership, and AccuBio. My path crossed with Ian in 2007 when he joined a startup company that I was involved with called Molecular Vision, and Ian was the CEO of that company for 3.5 years, tackling what one venture capitalist described as a 3-miracle problem. Needless to say, we didn't quite manage to solve all of the miracles, and the company eventually was then acquired by the Abingdon Health Group, and the technology was taken forward in some different ways. Following on from that, Ian worked for 5 years with Bestpac, Oxford Medical Diagnostics, and Arca Diagnostics, and he moved to Innovate UK firstly as the Director for Health and Life Sciences, and then as Director for Aging Society, Health and Nutrition, before then being promoted to the Executive Chair role. Given that this is a community event and KAUST is a community, I thought I'd also mention that Ian is very much a community-focused person, and so for more than a decade he has been the coach of Godmanchester Rovers Youth Football Club and also the Vice Chair of the governing body of the Stukely Meadows Primary School. Thank you. Ian.
Thank you. Thank you, Dono. And first of all, before I say anything else, I want to thank Dono for his personal invitation to come to KAUST. It's my first visit to Saudi Arabia. I'd also like to thank all of the organisers and actually everybody I've met so far. You've made me feel so welcome, and it's great to see such a young institute delivering such rich programs across a diverse range of topics in such a forward-thinking manner. So I'd like to say that before we get started. Secondly, I didn't think that my skills as a football coach need commenting on here, but needless to say, Godmanchester Rovers are a middle-of-the-league team, unlike Liverpool. So, Innovate UK is really the organisation that was formed as the Technology Strategy Board by the UK government in 2007. So a bit like COST, we're only a few years old. And it was devised at a time where the UK recognised it was very good at academic science, creating publications, creating patents, but its productivity was falling because we weren't given enough emphasis on building research and development into products and services. And so over the last 10 years or 12 years, we've been looking to how we can help the business community support great academia, become great business, and solve societal problems. But there's some interesting spillovers from that, because innovation actually drives productivity because it creates new thinking. A business that is innovative is more likely to be globally competitive and therefore increase exports and sales. And it also brings new ideas and new technologies to the markets in ways that maybe didn't seem obvious. So my role is really to create new value chains by funding high-risk, innovative research that results in business growth. Some statistics to back that up. So although the UK isn't great at scaling companies, we're actually pretty good at starting them. So we ranked 5th in the Global Innovation Index overall in the last year. Innovation itself accounts for approximately 50% of labour productivity growth. And what that means is as businesses learn and develop new ways of doing things, they become more productive and more reflective of the markets that they're trying to serve. Firms that continuously invest in research and development tend to have higher productivity by and large, and innovative companies are more likely to grow. So as the innovation agency, as Donal said, my job is really a glorified dating network. I like to find collaborations throughout academia and business that solve problems. So if an academic has a great idea that he wants to create a spin-out, I can fund them with some money to create a company that starts to do that. If an existing business is looking for a new technology, I can scan my database to find that to help them. Or, in a rare case where two businesses want to solve a common problem, but they can't do it themselves, I will help to facilitate that. It's both for economic growth, and I think more increasingly, for societal benefit and societal need, and we'll touch on that a little bit later. But to complete the evolution of Innovate UK, as I said, formed 12 years ago, in 2015 there was a review by Sir Paul Nurse. And Sir Paul Nurse said, we should bring all of the public funding of research together to make it more cohesive, more collaborative, and expect greater benefit. So the research councils, so including Medical Research Council, which many of you know, the Engineering and Physical Research Councils, etc., all 8 of them, combined with Innovate UK in the 1st of April 2018. Now, many of you may not know that the 1st of April in the UK is an interesting day because it's April Fool's Day. So we actually began on the Monday, which was the 3rd of April, but the company was formed on the 1st. And the real reason for UKRI is to develop knowledge with impact. And if we think about that from a preliminary research, that is taking findings that make interesting reading and can allow further development. When it comes to business, it's how do we create products and services, and ultimately, how does that benefit society as a result of those findings? One of the privileges that I get as the exec chair of Innovate is every year I have to go to the politicians and I have to say, this is my delivery plan for the year, this is what I'm going to do. And last year's delivery plan with the 5 themes that I've highlighted above was slightly different. Because we're part of the overall funding network for public funding of research, I decided to try and make us more collaborative with one reason in mind. I think that 2 heads, 4 eyes, 4 ears looking at the same problem will give you better solution than one individual looking at it on its own. So I really wanted to drive collaboration, and in particular, collaboration between businesses that need support and academics who have the current thinking that can provide it. So the first thing I wanted to do was create an organization that delivers significant impact, both economic and societal-wise, but also to support innovative businesses and individuals or entrepreneurs to be more aware of the potential that their impact could have in terms of growing and scaling, not just businesses, but themselves and their careers. Thirdly, to make best use of the landscape of the research that's conducted in the UK, which is globally competitive and world-class, and turn those findings into something new. Identify and support both existing industries and help to transform those that are competitively challenged, as well as create new ones, and then build a supportive environment, not just with funding, but with networking and connections to allow the UK to truly be a place where innovation prospers. So the good news is we are 9 months into the year. The bad news, I haven't completed all of these, and they'll take some time to do so. But I think we need to reflect where we've come from. So in 2007, the Technology Strategy Board was conceived. And over those past 12 years, we have put about £2.1 billion of public funding into the system. That has been matched by just over £5 billion of private funding. This is important because the UK wants to leverage private investment to increase the amount of GDP being spent on R&D. But that's resulted in about £21 billion of GVA growth, so that is jobs and products and services. Or put another way, we tend to raise about £7 for every pound we invest in a business. So those are the hard numbers, but actually if you look at the layer below, we've supported 11,000 projects, so that's almost 1,000 projects a year. Just over 8,500 organizations have benefited from working with us. And for each organization we've worked with, on average, we've created 9 jobs. So overall, I'm pleased to say, in 12 years, we've created nearly 100,000 jobs. What puts this in context is that we only invest in very early, innovative ideas. So to create 100,000 jobs from very early technology readiness levels is something I think that we should be incredibly proud of. It's becoming increasingly important politically that we need to align ourselves with current challenges. And I'll talk you through this slide. But I don't think there are 4 grand challenges. I think there are 3. I think aging society is a challenge that needs to be addressed. Many of you need to be reminded that you have aged since I started talking a few minutes ago, and that is going to progress for a long time. And what I think aging society means is how do we help you to age well, so that is keep you fit, healthy, continued lifelong learning, but also introduce employment prospects and keep society moving forward. We've got the challenges of clean growth. We have an aspiration in the UK by 2040 to stop using automobiles that are fossil fuel derived. But at the same time, we've also got to reduce our heating bills by 50%. 90% of our building infrastructure will be the same in 20 years' time. So therefore, how do we retrofit heat-efficient systems into our homes? And also, the workforce that we have just now represents 60% of the workforce for 2030. So as new jobs and skills come on in these economies, we have to train people to move into new careers. And finally, the future of mobility. I'm pleased to say I went on the autonomous bus this morning. And this is the second time I've been in an autonomous vehicle. The first time was at Heathrow Airport through a project we funded to move passengers to and from Terminal 5. And I have to say that by 10 years' time, autonomous vehicles will be commonplace throughout the world. And I think that presents challenges for the insurance companies, who will have to insure in a different way. It will also be different for drivers, who will probably have to learn to drive, but really not want to because somebody else can take the strain. So there are a whole shape of challenges around how we make that economy work. And as we move from the combustion engine, first of all, to batteries, and potentially maybe to hydrogen longer term, the bigger challenge there is that all of our manufacturing footprint for cars is based on having a heavy combustion engine predominantly at the front of the vehicle. Now, with batteries, you want to spread that weight, you want to spread that load. Companies are going to have to invest in new manufacturing technologies to build those cars and possibly make them of lighter, more composite materials. So we don't have our challenges to seek moving forward, and we'll talk about it in a second. However, underpinning everything that we do, I think, are 2 themes today. First of all, I was a biochemist. I studied biochemistry, as Donald said. And biochemists lived in a lab and looked at biochemical problems and came up with solutions, and that could be drugs or new crops or whatever. If we look forward 25 years since when I was studying, that problem is now a problem that includes physics, it includes chemistry, it includes engineering, and it includes data. So therefore, we have to become cross-disciplinary, multidisciplinary to do it. Secondly, there is no shortage of data. We create data every second of the day. How we use that data to make decisions is going to become increasingly more relevant, and I'll touch on digital pathology, where we funded an initiative to try and allow pathologists to concentrate on the 10% of the cases that need their expertise and leave the 90% of analysis of images to something that can do it much more routinely, so in the form of an AI algorithm. So I think AI and data crosses everything that we will be doing going forward. Back in 2017, we began to launch what we've called the Industrial Strategy Challenge Fund. It has two aims. The first aim is to make the UK more productive, and the second aim is to solve some of these societal challenges. And these are 4-year programs, and over the course of that 4 years, they will invest in projects, infrastructure, and skills. And the smallest program is about £20 million. The largest is about £268 million. And just touching on some of them in the health space, so for data to early diagnosis and precision medicine, which is the program that I was responsible for conceiving when I was in the health and life sciences role, we wanted to do 3 things. First of all, the UK Biobank is the best annotated collection of samples from healthy volunteers in the world. We made a pledge to sequence them. That whole genome sequencing activity will be complete by 2023. Secondly, we wanted to create digital innovation hubs, and that was to allow researchers access to the NHS data, to allow them to look at the data that's already within the system to try and conceive new therapeutics, new diagnostics, and new prognostics in a safe space where that data doesn't leave the firewall, but allows it to be exploited for both academic and commercial research. And thirdly, we are trying to create a whole AI suite of tools around digital pathology and radiology. There is a 24% shortfall of pathologists in the UK. A pathologist takes about 6 minutes to review a slide. 90% of the slides that they review are actually normal tissue. So how do we train algorithms across cancer, diabetes, and asthma to begin to paint pictures that we can then use the tools of tomorrow to make quicker diagnosis and then really utilize pathologist skills to develop diseases on their own? But I would say one of the benefits that I've got of being in my current role is I look across all aspects of these technologies. So for example, in clean growth, we're now looking at how do we, through the Faraday Challenge, create batteries that allow us to drive 400 miles. I have to remind myself that the reason why we want to drive 400 miles in one charge is because that's the same as a tank of petrol, not because we need to drive that far. On average, a car travels 20 miles a day in the UK, and yet the expectation is it should drive 400. So as well as trying to look at the societal impact of batteries as an alternative fuel, we're also looking at behavior and trying to understand, would we accept if a car needs charged every 100 to 150 miles? Secondly, transforming food is quite close to my heart because we've been challenged in the UK with doubling food production. The reality is, as a planet, by 2050, with the 9 billion people that Nick told us earlier are going to be existing by then, if we use and waste the same amount of food as we do today, we will need 2 planets to feed our entire society. That's not sustainable. We have finite water, finite land, so we need to have innovative solutions as to how we use not only farmland, but all the land to create food at scale that will allow us to benefit. And finally, with smart, sustainable plastic packaging, we're trying to work with big players to look at new materials for packaging, alternative sources to plastic, better reusability, and at the same time, new industries using new material for packaging that still allow food to be preserved for the same shelf life that we have today. So a wide variety of programs. But primarily, this conference is about precision medicine. And precision medicine, I think, starts with what is the medicine that we're going to have. So FaberX is a good example of a medicines manufacturing award. What we want to do in the UK is maintain our excellence in API manufacturing in particular. FaberX are using 3D printing to print pharmaceutical products for bespoke medicinal delivery. enabling truly personalized medicine. And we believe this is the world's first in-human clinical study using printless technology to formulate drugs that are bespoke for the patient. It was a £600,000 grant, a £1 million project, and they've been able to raise about £3 million of capital on the back of it. We also deliver infrastructure support. So we have a catapult network. Relevant to this audience are probably two. Medicines Discovery Catapult, because probably in over 40 years, the way that we're trying to discover medicine hasn't changed. We now have a full suite of discovery products that we can put to try and develop new therapeutics and new diagnostics that we need to look into more carefully. But the story I'll tell you about is the Cell and Gene Therapy Catapult. So in 2009, the Technology Strategy Board started their first collaborative research into regenerative medicine, as we called it at the time. By 2012, we realized that the UK had some great research in that, had some interest in small companies, but there were two main barriers: access to facilities and regulatory consent. So at that time, it took 2 years to get consent for a clinical trial for an advanced therapy. With the Catapult's framework and technical support, that's come down to 60 days. And at the same time, we've built a manufacturing facility that allows those companies to create small batches for clinical trial. And then last year, we launched the Advanced Therapy Treatment Centers to train nurses, doctors, clinicians, logistics and supply chain how to remove cells from a patient, take it to a laboratory, genetically modify it, scale it up, ship it back to that patient while it's still efficacious, and deliver it to give the therapeutic benefit. That is not packaging a white tablet, sending it to somebody and getting them to take it orally. So there is a whole supply chain around not only how we develop advanced therapies, but how the clinicians are able to deliver that at scale. And then hopefully over time, we can simplify the processes to bring the average cost of advanced therapies down. But as well as having catapults in the life sciences, we've also got high-value manufacturing, which looks at composites, it looks at additive manufacturing, it looks at nuclear de-escalation through taking rods out of nuclear cells and cutting them down and packaging them off safely for storage. We also look at carbon capture and storage as part of offshore renewable energy, and the applications for new compound semiconductors, particularly in the areas of quantum. But with manufacturing, there is a need for the UK to do something different. So monoclonal antibodies were developed in Cambridge and exploited commercially by a lot of American biotech companies to make them very rich. PCR was invented in the UK, it's made a lot of others rich, and we've also been good at creating a World Wide Web that has, again, made lots of other countries fairly wealthy. So one of the things that we think with physical sciences is if we can cite manufacturing and advanced manufacturing in the UK, then we can truly change the paradigm. So we've introduced the Medicines Manufacturing Innovation Centre up in Inchinnan in Glasgow. By the accent, this is not English. I have a Scottish accent, so I'm biased for this. But what we've done here is try to create some value in looking at how we could create small batches of medicines that could be regarded as personalized medicine. So dependent on your condition, dependent on your age, dependent on your prognosis, you might want to have more or less of a compound, and you might want that in combination with others. So the Medicines Manufacturing Innovation Centre is all about creating that flexibility to give new medicines to you in a quicker response time. Secondly, we want to really have a national consortium of intelligent medical imaging. So everybody's talking about AI. The reality is AI will only become real if we have lots of robust, standard, well-collated, well-curated data sets that allow us to do analysis at scale. So through the NCIMI consortium, that should drive digital pathology to be somewhat different. We also have in Glasgow, the eye care facility, so the Industrial Centre for Artificial Intelligence and Digital Diagnostics. So going beyond pathology and radiology, what other diagnostic devices would benefit from greater data integrity? And this is a partnership across academic trusts, across the NHS, and across companies. So we have multiple partners to try and ensure that we get a solution at scale for digital diagnostics. But also part of my role is to take ideas from small companies and allow them to thrive. So we came up with a concept called the Investment Accelerator. So for most health and life sciences companies, the value of the company is dependent on the active life of the patent. So how can I invest to allow the acceleration of the development to such a point where the patent is active for longer and therefore the product's worth more money for a substantial period of time? So we do that by combining our government funding with private equity. And so far, we've run a trial where we've spent about £75 million invested in 76 companies that have allowed them to raise just shy of £210 million. So that shows you that we can invest, again, at scale, and companies can benefit. Why do this? So if you're a company, and Donald will know what I'm talking about here, as a CEO of a spin-out company in the UK, I spent 9 months trying to raise money, 2 months congratulating myself that I'd raised money, and a month preparing to raise money again for the next 9 months. And all the while I do that, I don't actually add value to the business. So if we can get an investor earlier into the company, then that means that the CEO can concentrate on how he develops his business model and executes on the plan. The venture capitalists like it because if they come in earlier, they get a lower price, larger share of the equity, but they can also see a route to following their funding if the company does what it says. VCs are not geared up to do lots of transactions in a year, so therefore they use my independence and my assessors in order to do that technical due diligence, so they look at the company, the IP, and the market prospects and can make a quick decision, therefore accelerating the route to market for the products. So we have Seagram Diagnostics based up in Glasgow, or ClinSpec, and they have a novel marker that they're looking for using spectroscopic methods for brain cancer. By giving them an Investment Accelerator award of £260,000, it actually allowed them to conduct a clinical study with 400 patients and raise just over £2 million, all within 10 months. So that shows you that you can act as an accelerator and get follow-on funding, and that was really the aim behind what we were trying to achieve. We're actually best known for our grant funding. So those are programs that last between 6 months and 3 years and have a value of £50,000 up to £2.4 million. And what that does is that enables an innovative business to put an idea into our office, and then we evaluate it to determine whether or not that would be successful. The key here is to ask for the right amount of money, because if you need £1 million, you should ask for it. If you only need £100,000, then that's also what you should ask for. Be realistic about what you can achieve with the grant and really describe the problem that you're trying to solve and how we can work with you. And that, over the last 12 years, has given rise to 3 or 4 different unicorns that have started off just having a concept and going in through the smart funding route. The important thing is any award above £150,000 or greater than a year requires it to be collaborative. Again, going back to my same mantra, 2 heads looking at a problem are better than 1, and if you look at it from both an academic and a business perspective, you might get solutions you didn't expect. But Sirius Diagnostics were able to acquire £100,000 through the precision medicine round in 2018. They followed it up with a smart award of £400,000, and now they've just raised about £6 million recently, all in looking at ultra-sensitive, low-cost, high-throughput diagnostics in areas such as sepsis. So it really does make a difference in terms of getting their products to the market if they're a small company. Reprocell have also got a novel skin assay, so they essentially create a monolayer of different skin cells that you can test for toxicity testing to determine if a molecule is actually even worth taking beyond phase 1. Reprocell have received around about £1 million from Innovate, and they also work with the NHS and academia and Stratified Medicine Scotland. And the reason for that collaboration is important because without their access to the clinical data and without the access to the expertise in the Stratified Medicine Centre, they wouldn't be able to get the progress that they've made with their skin modelling technologies for toxicity use. The other area that we've been interested in working in is innovation loans, because we realise that a grant is good to promote a project. An investment accelerator is good when a company is looking to raise money, but an innovation loan is there when a company needs to take a step in one direction and it needs a piece of capital equipment, it might need to recruit some people, it might need to acquire a new skill. You can't do those with grant funding, you probably couldn't do it with equity funding, but an innovation loan would actually allow you to undertake a real meaningful change to your business. However, the word innovation is as valuable as the word loan because I cannot compete with banks. I am a government agency there to support innovation. So wrapped in the loan application has to be, how do you innovate and what is it that's going to be transformed by your business by undertaking this? So over the last 18 months, we've given out just shy of £100 million and allowed companies to develop in strange places. So I don't know how many of you know the UK, but Exeter is a small town on the south coast, and no company ever in Innovate UK's existence had been funded if they'd been in Exeter until the loans. Now we've been able to fund 4 companies in the creative industries that typically wouldn't have been supported through our activities to develop new products and services to allow them to get to market. But innovation loans in themselves allow businesses to scale, but the word loan is important. We do expect it to be paid back at some point in the future. An example of this is Aston Particle Technologies. Now, they're based in Birmingham, and a spin out of the School of Pharmacy there. And they're an interesting case study because one of the challenges in the UK is, I've got a great idea and I want to create this company. So we've got a program called iCure, which is the commercialization of university research. And we give the individuals 9 weeks to answer a few questions to tell us, are you a company, are you a licensing opportunity, or is this needing further fundamental research? So if we complete that first body of work and they say, actually, we think we're a company, we then ask them to do some more homework, and that is, tell us who your CEO is going to be, tell me who your competitors are, tell me what market you're going to go into, tell me how you're going to be competitive in that market and what price you think you can make your technology for. If they pass all that, I then give them £500,000, and that allows the company to start from a sure footing of being able to have some seed funding and begin to develop their concept. So Aston got an iCure award. They then got a collaborative research award to progress towards full development, and they also got a loan. So they had all the suites of products and have now gone to market with their novel dry particle coating technology specifically for processing drugs without the use of heat or solvents. So again, looking at the societal impact of that, they are taking manufacturing efficiencies into account, they're more productive, and they're getting novel compounds into the clinic. So it's quite a good example of how our suite of products come together to help support the company. One of my predecessors said that Innovate UK's role is to talk to people, to connect to people, to try and do great things with companies, and if all of that fails, we should give them some money. So we come up with the Knowledge Transfer Partnership, and the key there is knowledge transfer. So we have to link UK businesses within the ecosystem to allow them to work with academics, to work with other businesses, to build supply chains, to identify markets. And you can do all that if you're a well-funded business, but you don't know the right people to connect to. I say that innovation is a contact sport, so you have to know the right people at the right time. You have to be fortunate to make those connections to allow you to progress. So what we try and do with the Knowledge Transfer Partnership is allow academic ideas to flourish through connecting them with the right people. But also, if a business has a technical problem, you can apply for funding to take an academic into your business to solve that problem. The programs can last from 12 to 36 months, and the whole idea here is the business benefits from current thinking, The academic benefits from going into the business and understanding more about the commercial world and whether that's a career for them, and overall the company gets more creative solutions through employing them through that period of time through access to the collaboration links. One example of this is a company in the UK called Storm Therapeutics, who are a spin-out from the University of Cambridge, but they actually had a problem in mining data. So they developed an analysis tool called Intermine to help them really map complex biological data, making it easier to query and access information from it. And the aim of Storm is actually to develop specific cancer therapeutics. It wasn't to develop a database that really mined biological data, but the creation of Intermine has allowed them to sell that as a product, but it's also advanced their cancer therapeutic targets. I was pleased with some of the comments earlier about diversity, equality, and inclusion. And in 2018, we did a review that showed that only 12% of lead applicants into Innovate were female. And so we inquired as to why that was the case, and it really fell into 3 buckets. First of all, there was no peer-to-peer network to allow female entrepreneurs to work. Secondly, a lot of the pitching that they did was at male-only panels. And thirdly, the route to capital for them wasn't always obvious. So we decided to tackle this head-on by launching a Women in Innovation program. We created 24 female leaders that were each funded with £50,000 and each given the benefit of some financial support, some mentoring support, and some networking support. And I don't know how many of you have been to London, but on the walls in some of the London houses, there's a blue historic plaque that tells you Winston Churchill lived here or whatever. And there are thousands of these plaques through the UK. And only 12% of them are for females. So I thought that's a bit of a coincidence. So what we then did is we told every Women in Innovation winner that we wanted to put a purple plaque on a school of their choosing to remind the school children that it's all right to have aspirations to be entrepreneurs. So I'm pleased to say that 100% of the purple plaques are dedicated to female Women in Innovation winners. But the second story I'll tell you is about Ideas Means Business. So believe it or not, for you 18 to 24-year-olds out there, research would show that innovateuk.gov.uk was not the highest priority in your search engines. And so we thought that we're missing out on a valuable, valid set of society that has entrepreneurial aspirations and wants to go businesses, because actually 60% of young people under 25 have a business idea. So our job as Innovate UK is to try and create economic growth. So we said, okay, we'll run a program for young innovators. How are we going to do that? And we puzzled for a while. Then we said, okay, we'll try and use the Prince's Trust, who are a charity in the UK, for young people, and particularly to help homeless youngsters. So in thinking about how we launched the program, we did 2 things. We said we're going to launch it on Twitter through the hashtag #IdeasMeansBusiness. And secondly, young people in the UK are also very much affinity with the coffee shop culture. So we had pop-up coffee shops around the UK. And a gentleman called Adam Root was a successful winner of an Ideas Means Business application. Now, Adam has a plastic microfilter that fits into a washing machine that allows you to capture the microplastics so that they don't go into the food chain. And Sky News were really excited and wanted to speak to Adam, so we got a phone call, could we reach Adam in order for Adam to speak to the press? Nobody had Adam's number, no problem. Adam must be on Twitter, I'll reach out and get him on Twitter. Adam also didn't have a Twitter handle. What we discovered is that when Adam was in Cardiff for the day, Adam liked a cappuccino. So serendipitously, he walked into a pop-up coffee shop and found out about our competition. Subsequently, he's raised just about £2 million. He's about to conclude a deal with a white goods manufacturer worldwide, all on the basis of serendipity, because if he hadn't walked into the coffee shop, he wouldn't have won the money. So therefore, my other tip to you is, it's good to be good, it's also exceptionally good to be lucky. So bear that in mind, that serendipity is still a valid scientific discovery tool. Finally, I'm going to say that if you want to have a business that wants to grow, you are going to have to meet the right people. You're going to have to meet the right people, and you're going to have to meet lots of them and tell them about your idea. One of the things, and I'm sure Donald will support this, is time after time, you have to go out and meet either fundraisers, supply chain people, manufacturers, investors. No matter who it is, you've got to meet them. So I would encourage you to be bold, be brave, and remember, you know more about your idea than they do, so you always have the upper hand. I wish you every success. I hope great ideas come out of KAUST. Thank you very much for your time. You're welcome.
Okay, thank you so much, Ian. So the floor is open for a couple of questions. I already know I have a few of my own, but I'm just wondering if some of you would be interested in having some more insights about the innovation culture in the UK and how they fund startup companies and how they encourage students and researchers and entrepreneurs to actually invest and spend some time and take some risks at this early stage of their career. Okay, questions? Okay, so my question is, how do you judge that this is a good idea, that I should fund it or not? I mean, what's your go-to key thing that personally, I mean, there are criteria, there are rules and tables, but personally, how would you treat that?
So I think there's two elements to that. So first of all, we're fortunate. So every application sees 5 assessors. And they are a combination of business people and academics, and they are looking at how good is the idea, how likelihood is it to succeed, how clear is it in the proposition what's going to be done through the course of the grant. My one big tip is be clear and be concise, that you understand the problem that you're trying to solve, and try and write it in as few words as possible. It's always easy to write 60 pages. It's not so easy to write 2. If you can't get your idea onto 2 pages, it's probably the wrong idea.
Thank you so much, and please join me in thanking Ian for his wonderful talk.
Thank you.
So it's not easy to be the last speaker for the day, so bear with me. So we have a nice exhibition in Building 19, which is across from here, where we have a collection of Saudi startups, international startups, mostly from the UK, international companies, all are in the healthcare area. They are looking into personalized medicine, diagnostic devices, AI, machine learning, how to actually figure out some techniques to improve the quality of life of patients. With that, I'd like to thank you all, and I'd love to see more of you attending our events. Tomorrow we have 2 keynote talks, and we have our first culture event, which is a jazz concert, and this would be in the evening. So please try to enjoy all these events and try to attend as many as possible, because you never know, as Ian said, who will you meet or who will you be sitting beside in any of these events that could be your future partner, future employer, future boss, future investor. Try to talk to each other and learn from each other. Thank you so much.